This is a production of Cornell University.
So good afternoon, everybody. It's kind of nice to be back, I guess. It's been a couple years. I was hoping to get home this winter, but we'll present this stuff remotely today. Please, again, feel free to ask questions. And Marianne, please interrupt me if there are questions that come through, and feel free to, I'd rather have a conversation than just talk to my screen. And before I get started, I'm just going to share some resources that we've developed out here in Indiana that are available. So if there's anything I present, if you want to look further into that, you could look at some of the diseases we're monitoring and some of the publications that are out on the various diseases. I'm going to try to give a sweet corn angle, but now that I'm at Purdue, we're mainly focusing on field crops, but I think we can talk about plant diseases the same way and try to adapt them for sweet corn. I'm going to get into a little bit on tar spot just to give you an update on what to keep an eye out at the end. But first, I really just thought I'd start about, what really leads to disease developing. And we, as pathologists, like to talk about the disease triangle, but I think it's really important to understand the three contributing factors that can cause disease. First of all, is our susceptible host there? So we're talking sweet corn, what diseases affect sweet corn? Is there a growth stage where the diseases are going to infect and cause problems? Are there hybrid or variety susceptibilities that'll also increase the risk for that host? So that's one side of the triangle. The second side of the triangle is, what pathogens, what diseases are we looking at? How are they surviving year to year? So if we're thinking about northern corn leaf blight in New York, it does overwinter in the debris, so it's surviving year to year. So it's important to understand if that field has had a history of it. It may be present in those soil, in the soil to cause a problem. There are other diseases that aren't overwintering. For example, many of our rusts, we monitor southern rust, and common rust would be an issue in sweet corn. Those spores don't survive, so then we monitor the spore movement and when does it move in to the state to cause problems. Is it moving in early enough in that season to cause an issue where it may reduce quality or yield of that crop? And then finally, the third factor is the environment. And environment is the key of telling us the risk for that season. Every disease has its own set of environmental conditions, whether it's temperature, moisture, leaf wetness requirements. For example, if I'm talking about southern rust in corn, it only needs 4 hours of leaf wetness for that disease to continue to cycle through its life. Whereas when we get into tar spot, we know it requires a lot of moisture. 8 to 10 hours of leaf wetness are required for that disease. So again, if the environment's favorable, then we increase our risk if the host and pathogen are present. So if we have an unfavorable environment, maybe we don't need to be implementing tactics this season because the environment isn't going to push that disease along. But as those conditions become favorable, that's when we increase our disease risk. And so once we understand what diseases we're looking at, there are generally 4 main areas that we can manage disease in corn. The first and foremost is looking at genetic resistance. Do our varieties have resistance to some of those diseases we're most concerned about? Gray leaf spot, northern corn leaf blight. Again, we're acting on that host part of that disease triangle. We can also look at crop rotation and sanitation. And the idea is if we don't plant a crop in a field that's had a history of disease, we can reduce the inoculum that's available. We remove that host from that environment. So again, we're reducing the host and pathogen aspects of the disease. And sanitation wise, if we're thinking about it, we can till the debris under the ground and have those microbes work on some of that inoculum, reducing that initial inoculum, maybe reducing that initial risk for the disease. We can also talk about, mainly our soil-borne pathogens. Are we moving that inoculum between fields? Maybe there's a field that's got a bad problem. We don't want to be moving that diseased soil into a new field that may not have that problem. So again, we can think about sanitation and trying to reduce the inoculum and the spread of that pathogen. Again, acting on the pathogen part of that triangle. And then finally, if a fungicide is necessary, we might make a pesticide application, and that's going to act mostly on the pathogen side of things. But as you can see, the one factor we can't control is the environment. And so every year, depending on the season, it's, we can't project what the weather is going to be in June yet. But again, that environment's what's really going to dictate how severe or if there's a tactic that needs to be implemented for managing diseases in corn. And I first wanted, we tend to use more fungicides out here in Indiana and trying to control some of our diseases, gray leaf spot, northern corn leaf blight, and now tar spot. And I just wanted to review a little bit of what our fungicides are designed to do, because especially with this new disease, we're seeing that there are limitations of what what they can do and how much they protect. So our fungicides are those synthetic or natural compounds that help protect the crop. They're designed to kill or inhibit the fungal spores or the spore germination on that plant. Generally, we have two groups of what we call fungicides, those that attack the true fungi, the chitins, Rhizoctonia, fusariums. They're going after the fungal and cell walls of that. There are also the cellulose attacking fungicides. They're going after our fungal-like organisms, the oomycetes like the Phytophthora, Pythians, and downy mildews. So it's important to understand what disease we're targeting, what that pathogen is causing the disease, and then help us select what fungicide target we want to go after. When I look at field crops, we generally have three modes of action in most of our products. So we have the strobilurins, the group 11s, the DMIs, the group 3s, and the succinate dehydrogenase inhibitors, the group 7s. Generally, we have various ratios of these products available to try to inhibit spore germination and infection across the crop. The one thing I want to note, if you look at these, all three of them do have a more targeted site activity, which also means that there is the chance down the line, if we can continue to apply the same fungicides over and over again, we can lead to disease resistance in those crops. So that's an important thing to understand. But generally, when we're looking at corn, there are three modes of action. In sweet corn, you also have the broad spectrum with chlorothalonil to protect the crop that you can rotate in as well. So, but it's important to understand, maybe looking at these products, if we're applying multiple times a year, maybe rotating what products get applied to the field. And the other thing I just wanted to review is the stages of fungal infection in a leaf and how we try to target to minimize disease. So if you think about a fungal disease, the spore will land on that leaf surface. It begins to penetrate the cell walls either directly or indirectly. And then once it breaks through that leaf surface, there's a mycelial growth stage. That's kind of what we see when we see a Petri plate and we got a circular growth of the fungus growing. And then it moves into a developing its structure to release more spores and complete the life cycle. So some kind of sporulation structure will develop, and then it will release those spores, and the cycle can continue. And depending on the disease, the cycle can take 5 to 14 days from spore to spore. So again, environment and pathogen are going to dictate how many cycles we have in the season, and depending on that will determine how fast the disease may develop within the crop canopy. And if we look at how our fungicides are designed to reduce disease severity, it's important to see where they're attacking that disease, the fungal growth cycle. So if we look at chlorothalonil, our broad spectrum fungicide, it's attacking the germination of the spores. This is a protectant, so it needs to be down before the spores land on the soil surface. Our strobilurins are acting on spore germination and penetration. Again, generally they need to be down there before that pathogen's moved into the cells of that leaf surface. The triazoles, they do affect the penetration and mycelial growth of the pathogens. But as you can see, in general, if we're looking at these three groups, we got to get ahead of that spore germination and the penetration to slow it down and try to limit that disease cycle that may occur within the crop canopy. And so if we look at that, how the life cycle of a fungus grows, the QOIs and the SDHIs are designed to inhibit that spore germination and penetration, whereas our DMIs are inhibiting that early fungal development. This is, again, this diagram I showed earlier. The QOIs, the idea is that they need to be there preventatively. They need to be down on that leaf surface and available to slow down those spores before they land there. The DMIs may be giving a more curative action of controlling the diseases, but in plants versus humans, if we take a curative antibiotic to slow a disease down that we may have in humans, we kind of cure the disease from us. The problem was in plants, we really can't backtrack if we've lost that cell area to the disease. What they're doing is the curative action is it's slowing down what's already been infected and stopping it from continuing, but we can't just turn disease tissue back green again. And I think that's really important, particularly with corn. Once we get to reproductive stages, we're not adding any leaves to the crop canopy. So we need to make sure we're protecting that ear leaf and above to really get the optimum yield and benefit from that crop canopy. But if we have diseases move in and they're taking away that photosynthetic area, we're not going to be able to add more leaves to replace what is lost. So that's really important. And some of these diseases, particularly when we're talking about tar spot, is we got to get ahead of the disease to slow it down in order to prevent the yield losses that could occur. And so when we talk about fungicide timing, particularly on field crops, the idea, even corn, soybeans, whatever crop, is, we need to be out scouting. Is the disease active in that lower canopy? Should we make a decision to put a fungicide out? And then when we put a fungicide out, we have a residual protection. Generally for our fungicides in field crops, we have 2 to 3 week window of activity of those products. So the idea is we want to put the fungicides out when we know the disease is going to be active, so we get the protection we need, but understand after 2 to 3 weeks that fungicide is going to run out of activity. Maybe in another field the disease moves in later, we can put a fungicide out and hold on to it and apply it later in the season. But the idea is we want to lay our fungicides when we know they're going to protect the crop. If the disease doesn't move into the end of the season, maybe it's not going to have any effect on the production and yields of that crop, or the quality of the product that we're trying to harvest. But again, the idea is we're going to spray our fungicides when disease is going to impact yield. And if we look at just a little bit at what gray leaf spot does in grain corn, just an idea of when we lose that photosynthetic area, how that leads to yield loss. And so this is just some work that's been done in the late 90s looking at gray leaf spot. So if we can keep the ear leaf and above green to 5% or less disease, like this upper picture here in the corner, then we're going to have very little yield loss occur. But as we continue to lose that ear leaf to the diseases, that's when we start seeing 2 to 10% loss, or up to 50% loss if we lose those leaves that are leading to the production of those ears. So it's just important to understand how much yield do we protect, and does that give us a return of putting a fungicide out. So when you get ready to try to make a selection of fungicides, there's a very useful tool, and the 2022 data has just been released. We take disease data across the country and we contribute to this efficacy control table. So this is looking at all the various corn diseases across the United States. So the main ones that we're going to manage. So for here in New York, northern corn leaf blight, common rust would probably be the two that you're looking at, maybe gray leaf spot or anthracnose. But you can use this tool as an option to try to identify what products are available, how good they are at controlling the various diseases, and then kind of give you an idea of what you could target for your farm. If you have 2 different diseases, maybe you can pick one that has a good rating for both of those diseases. But again, a tool to help you try to identify what products might be available. You're going to have to cross-reference to see if it's labeled in New York. And particularly for sweet corn production, there's a good part of the table here that can give you the harvest restrictions, so you can understand when the fungicides can go out and how soon after you can harvest for the various crops. And so Marianne asked me to cover a couple of diseases, and I just thought I'd review some of them and give you an idea of what we think about how do we manage them. So Stewart's wilt was one disease that she asked me to cover. We generally don't see too much of it here in Indiana, but again, it depends on the year. So this disease is spread by the corn flea beetle. And depending on how much a flea beetle survives over the winter, how cold our winters will dictate the population of them coming out and feeding on the corn. The flea beetle will spread this bacteria, and then once it feeds, you start seeing these water slope lesions to develop. And if we get a significant infection, or if you have varieties that are susceptible to flea beetle, that's when we might start seeing some severe wilting occur in sweet corn. And so when we look to try to manage Stewart's wilt, there is some useful information on the monthly temperatures that are available. So if December, January, and February are warm, then we're increasing the survival of the flea beetle over winter. That means there may be a higher potential of this insect to come out and feed and then spread Stewart's wilt through the corn. So again, you can see there are various stages where it can lead to a seedling wilt stage. Here you can see some of those leaves burning and plant death if it gets early enough into the corn. To manage Stewart's wilt, generally you can look for tolerant sweet corn varieties, and then also make a decision if we know we've had a warm winter, maybe it's maybe a seed treatment for flea beetle management might be necessary. So that's one disease where we're not going to manage it with fungicides, but we can look at managing the insect that may be spreading this disease. The other disease, common rust, we generally don't worry about it in grain corn, but I know it can be a problem on sweet corn and reducing your quality. So we do try to distinguish common rust from southern rust. I suspect in New York, you generally see common rust. Sometimes here in Indiana, we do have southern rust move up. So it is important to try to distinguish the two. Common rust, it has these pustules here where they're rupturing on that leaf surface and releasing more spores. The pustules can form on both the top and bottom of the leaves. So that helps us distinguish it from southern rust. And so looking at this life cycle for common rust, we know that the fungus is overwintering in the subtropical regions. The wind is carrying those spores up from Central America. They're going to blow into the United States, and they're going to land in that crop canopy and cause these pustules. Depending on the season and how early they land will dictate how much of that disease continues to develop within the crop canopy. So if it comes in early enough in sweet corn, it may justify a fungicide spray to try to minimize that. And then at the end of the season, they're going to produce these black pustules, and the spores are going to be moved back down into the subtropical regions. So that's kind of the life cycle that we see for common rust. And so again, to manage common rust, again, it likes cooler temperatures. We generally are scouting vegetative to early reproductive stages to make a decision if a foliar fungicide on sweet corn is necessary. You can find those hybrids that are less susceptible to common rust. The other disease I know you see a lot of in New York is northern corn leaf blight. It produces these classic cigar-shaped lesions on the leaf. Here on the right, you can see them. If you had a hand lens, you might be able to visualize the conidia that are forming within those lesions, and we identify them with these lines of a conidia that can form. You can also send samples in to confirm to the diagnostic lab. When we talk about northern corn leaf blight, we do know that this fungus overwinters in the debris. So fields that have had a problem with northern corn leaf blight, if we're rotating back into sweet corn again, may have a problem this season if they have those environmental conditions that are conducive. So we know wind and rain spread those spores up into the crop canopy, and then you'll begin to see these lesions developing within that canopy, and they can occur anywhere within the canopy depending on where those spores were splashed up into the crop. You're going to have larger lesions in those high varieties that are more susceptible to northern corn leaf blight, whereas those that may have some resistance may show some small lesions where they've inhibited the development of that disease within the crop canopy. The cycle can continue through the season, and then that infected debris would then be laid down and available for future seasons as well. When we look at northern corn leaf blight, again, moderate temperatures, wet and humid conditions. So if we have a wet season like we did this last year, that could lead to more northern corn leaf blight development. Generally, there are many of our varieties in sweet corn do have a rating for northern corn leaf blight. So you can look at those varieties that may have a higher resistance to it. We look at crop rotation as a way, again, to manage that debris and the survival year to year. And then again, you can make a decision on putting a foliar fungicide out. And then finally, I just want to give you a little update on tar spot for a disease that you need to be keeping your eye on in New York. It's not, it hasn't been there long enough. I know Gary had his first finds at the end of last season, but it is present in New York. And I just want you to be aware of this disease and be on the lookout. It will be important to track it over time. When I got here in Indiana, this disease landed on my lap, and I've pretty much been spending 80% of my time trying to figure out how to get ahead of this, where we're seeing significant losses here in our grain corn in Indiana. But when you're out scouting your corn, the first thing you need to look for is these black stromata. So you can see here on the left, this is the earlier infection of the disease. You can see we can get multiple infections. As we talked about the life cycle of our fungi, this is the spore structure. So this is the end part of that life cycle. It's where the structure's there. It's going to open up and release spores to continue on the secondary structure. What we can't see is that infection that's occurring, that initial infection and the colonization of that leaf. This pathogen is an obligate pathogen, which means it requires a living host, just like our rust. So we can't really easily work within the lab. It requires green tissue. So our green healthy corn is what this disease is going to go after. When we get severe infection, we can have this rapid change from the left picture to the right picture in a couple weeks here in Indiana. And that's what's been surprising about this disease. is that it's not as slow as northern corn leaf blight or gray leaf spot. Once it gets going in the crop canopy, we have the severe infection and we have the severe blighting that can occur. And here in hybrid corn, at least if this happens before dent, we can see significant losses. So depending on when this disease moves in sweet corn, it may or may not be an issue. I think it's probably going to be an issue for any of those later plantings if the disease does ramp up within the season. And so this is just some images of the various symptoms that we see with tar spot in Indiana. We have only identified the one pathogen. It was first found in Mexico over 100 years ago, and they talk about a complex of 2 pathogens. But here in the Midwest, we really have only identified the one, the Phylocoramatus. But we do see the symptoms that they talk about where the secondary pathogen moves in, this fisheye lesion here that does occur once in a while. It's not as readily seen as much as just we have these black spots. But we have not found the secondary, and we've determined that we really don't need the secondary to cause significant losses here in Indiana. But you can also see that it does survive in their debris. And we've done work with our counterparts in Wisconsin and Michigan to show that yes, the spores are surviving year to year in the debris. So we have, once it's here, the inoculum's present and it will be available for future seasons to cause disease. And so the one thing about scouting for tar spot here in Indiana, at least, is it's the first to find this first stromata, which is this black tar spot, is really hard. It's a needle in the haystack, but it has been important to try to determine when the disease is first active, and that will dictate what management tactics we may need to implement in season. So this is just an example. Usually it's the 4th full leaf open at my knee level is where I've been finding it. But this, initially to find this, we look at it, it's embedded in the tissue. So you scratch at it, it's not going to come off. You can actually see it grow through on both sides of the leaf. And then as it develops, that's where the spores are going to be released. The thing about this disease is that was 1 week. I went back a week later and we easily found tar spot. So it changes relatively fast once it gets going in the crop canopy. So we can go from 1 to hundreds of lesions in that lower canopy. And then it will further develop as it moves up the crop canopy. And so we do track tar spot nationally. So this is the map from 2021. And so in Indiana, generally we see it come out about mid-July is what we were first predicting. This last season in 2021, it was the July 3rd weekend when we found it both in Indiana, Michigan, and Ontario, Canada. But as you can see, it's continued to spread. So this is the map at the end of the season. So you can see it moved through Ontario, Canada, found it last year at the end of the season, and they got hit really hard this year in 2021. And you can see they also had a first find in New York at the end of this season. It wasn't severe, I don't think, but it is now present. So it is something just to keep an eye on what happens. And then also Georgia, Kentucky, and Nebraska also had their first finds for the season. So it continues to spread. I think this next map shows where we found it over time. So you can see in Indiana and Illinois, we found it in 2015 and 16. We thought as a cosmetic disease, it really wasn't causing any significant losses. But in 2018, we had the perfect environmental conditions. So we were building up inoculum slowly year to year. It was surviving every year. We were seeing it expand a little bit from 15 to 18. But in 18, we had the perfect conditions. And you can see that large pink area where we had significant impact here in Indiana, Michigan, Wisconsin, and Illinois. We documented about 20 to 60 bushel yield loss is what we estimated at the end. 19 and 20, we continued to see it spread. You can see it move across Iowa. They're the first state that confirms that they have it in every county, but their risk depends on the environment. And then in 2021, I would say it was just a worse year than we had in 2018. So the environment we had a lot of moisture conditions that led to some severe losses. I had growers that lost 50% of their yields from this disease. So it is an important disease to keep aware of. I just want you to know to watch it. It may not quite be impacting New York yet, but no, it might be coming your way. And the things we know about the disease is, again, we know it's overwintering in the residue. And then once in that spring, when we have the right moderate temperatures and a lot of leaf wetness, it's releasing those ascospores. And from that black structure, the stromata, Those ascospores are released forcibly up into the crop canopy so we can inoculate all the plants in the field. And then usually we see symptoms about 14 to 21 days after infection. That's kind of what we're estimating from what we've worked on in the greenhouse. And then that cycle is going to continue within the season. That secondary cycle seems to be much faster than that first infection period. And we're still not sure how fast that cycle occurs. But I know in our field plots that I can see the corn changing day to day on the amount of infection that occurs in our crop canopy. And then the diseases will be returned and dried to that field for future seasons. And it's important to note that in Indiana, we have a shorter rotation than you do in New York. We're generally corn and soybeans, but I've been in fields that were soybeans in the previous year, and the corn probably 2 years prior had to have had a little bit of tar spot for the epidemic that started in the next season. So it is important to understand that it may not be the next year. It could have been the infection from previous years that led to the disease that was coming into the corn for that season. And so in Indiana, we tried to make a real effort and try to look at the risk across the state. And so by the end of August, early September, we get out and try to get into 2 fields in every county to try to see how easy is it to find tar spot, and then how severe is it in that field. And so I can turn a county positive initially with just 1 stromata on a leaf. That's not really going to cause any yield losses. It's when we get above this 5 to 7% severity, we might start seeing some impact on a yield. And we see that severe blighting when we get above 25% of those stromata, that leaf just being completely colonized by the phylochora, the pathogen of tar spot. So we get out and we rate how many plants can we find tar spot on and how severe is it. And from that, we came up with an index. And so it's taking the incidence by severity. And this is just to show you the changes that we're seeing here in Indiana. I think as we continue to build up inoculum year after year, then I'm starting to see more pockets develop. So in general, it was a lake effect problem. So northern Indiana, off of Lake Michigan, where we're getting the weather and those long periods of dew, that's been the area of the hot spot. So even though 2019 and 2020 were drier years, we still have a region up in northern Indiana that's consistently seeing a problem after that first epidemic in 2018. In 2020, we kind of condensed the hot spots. So those red areas are condensed because we had drought conditions in the state. But as you can see, we're starting to see where we see more and more inoculum, more plants that have the disease in the south and central part of the state. And then in 2021, we had very conducive conditions for tar spot. I suspect the top third of my state, probably if they didn't implement a good hybrid or a fungicide timing, probably saw 20 to 60 bushel yield loss. You can see in some of the counties, though, that my color is a little lighter. So that tells me whatever field I got into there, either they had found a hybrid that had reduced the severity of tar spot, or had put their fungicide out at a right timing to limit the disease. I suspect every plant in northern Indiana had a level of tar spot or not. But the surprising thing for this past year was that I have this hot spot down here in southwest Indiana. And if you look at the southern Indiana there, it's along a river. It's the riverbed area where this disease ramped up. And it came out, it was one of my first fields that had a severe infection this season. So again, the inoculum had been there for a couple years, and then the conditions were right for it to take off in some of these fields. So that's something that's really important to understand, is it may not be a problem for a couple years, but if you build up enough inoculum and then have the right weather conditions, this disease can become a severe problem. And so when we'll look at the drought conditions from 2019 to 2020 in July and August, you can kind of see where we slowed the disease down. We had some droughty conditions those last couple of years when we were surveying it. 2021, we moved in and we had a wet July. And so the whole state had been cleared as drought monitor conditions. But by August, the center part of my state was experiencing severe drought. Their worry was not having enough water for the corn. I could find tar spot, but it was not an issue in that band across that part of the state. So again, back to the disease triangle, moisture is what's driving this disease. So this is an image from my counterpart, Marty Chilvers, up at Michigan. This whole field had been treated with headline Ampid R1, which was one of our products that does work for tar spot. But again, what we're finding is the environment may be so favorable that you're still going to have a lot of disease and reduce, everything we throw at it still may not reduce its impact. Now, depending on the season, is dictated whether these green corners out yielded this under the center pivot. For 2019, I believe still, even though we have severe tar spot under the severe pivot, they still had better yields under the pivot. The crop needed the water that year. In other years like this year, many of my guys that have irrigation up in northern Indiana, their green corners yielded 100 bushel or more better than under the center pivot. So again, water drives this disease. If we give it a little extra water, it may push it to where we have yield losses. We're still trying to figure out how to best manage our irrigation, where we can give the crop what it needs, but not push the disease through. But it's important to understand, at least here in northern Indiana, we're going to continue to see tar spot from now on until we can develop more resistant hybrids. And so looking at 2021, it was a bad year for tar spot. And this is looking at the precipitation departure from June to August. And you can see there are some bands that are very interesting on where the problems occurred. So my counterparts in Wisconsin really didn't have problems with tar spot in 2019 and 2020. They had 2% or less disease. Whereas in northern Indiana, around this Lake Michigan area, we've consistently had, yield losses every year. But you can see that Wisconsin band, that is where they had severe tar spot this year. In northeast Iowa, that spot right there is where they saw it. And if I had Ontario, Canada, they had 6 to 10 inches of extra rain in June and July, and they had severe disease. So water will drive this disease. And so that's the biggest concern. And if I look at New York, you guys have those conditions if you get enough inoculum there. And so the next question comes, and let me know if I'm running out of time, managing tar spot. So we've been doing a lot of different trials to try to look at how do we manage this disease in our row crops. The one thing, first of all, is looking at susceptibility. So this is just an example of where we looked at susceptibility to tar spot back in 2018. As you can see, we have hybrids that have low disease severity. They can keep it under 1%, whereas we have those that are, have 50% or more. And that is a difference between green corn and blighted corn in the field. So this is just an example of two hybrids next to each other. Both are infected with tar spot, but you can see that one is just staying green longer, trying to get to its natural maturity to hold on to that yield. So we tried to look at the impact of tar spot on our yields, and what we were finding is we lose about 0.3 to 1 1/4 bushel per 1% increase of that tar spot severity in that ear leaf, about 1/2 bushel for our early maturing varieties, and then 0.8 of a bushel for our later maturing varieties. So again, there is a range of susceptibility of our hybrids, and there's probably a range of susceptibility in your sweet corn varieties as well. We did try to look at layering hybrids by fungicides on tar spot. So this is 3 years of data, and where we had a susceptible and moderate resistant and 2 moderate resistant hybrids out there to look at, can we reduce the amount of disease in that crop canopy, and then will added fungicide help give an added benefit to it. So here we're looking at, we treated with Trivapro at the VT ditassil to R1. And you can see where we had the untreated, what we can see is that we do decrease the disease with adding a fungicide across all 3 hybrids. But then the question comes into play, is that decrease enough to give us a yield benefit? So yes, we can reduce disease using a moderate resistance, and then we can further reduce the disease by adding a fungicide on top of that. But when we look at the yield benefits, we don't see that increase in yields is significant with our resistance. So some of our resistance packages may provide us enough, may slow down the pathogen enough to protect that yield potential. When we have a susceptible hybrid, we definitely can see an increase in yield by adding a fungicide. And this was at using Trivapro at BTR1. There have been many other questions on what other factors can increase disease, and one of them looking at residue. And so this is some trials from 7 trials that have been run in Wisconsin, Indiana, Illinois, Michigan, and Wisconsin, looking at tar spot on high residue versus low residue fields, and then looking at resistance and susceptible varieties within those. And so what we're seeing is, yes, with a lower residue field, we can reduce the severity of tar spot, those blue bars in that corner, we can reduce with a lower residue. So if we can plow on some of that initial inoculum, we can slow down the disease a little bit and reduce the severity. If we look at resistance here in the green bars and versus susceptible, yes, our resistant varieties can significantly reduce tar spot severity over our susceptible hybrids. If we layer them together, what we see though is that resistance. So if we look at high residue and with a resistant and high residue with a susceptible, we get a bigger bump in reducing tar spot severity when using our resistant hybrids, versus it's not quite as big when we look at high residue versus low residue. So that is an additive factor, but again, resistance is that's one factor that we really need to target in trying to manage this disease. The other thing that we've noted is we generally see the disease ramping up about R3, R4, so milk to dough, and then it takes off by dent. So in sweet corn, you may get away without it really significantly impacting yield because you'll be harvesting at that point. And so it may not lead to some as significant yield losses. You might lose quality, I think, if you get infection on the husks and you're selling within the husks. But again, I think possibly in sweet corn, if we do see this, continue to see this trend on the growth stage, you might be able to escape some of that infection. I do worry though, once we get severe infections going and we have a severe epidemic, there's a lot of inoculum out there. So for the later planted corn is going to have a lot more inoculum and affecting it earlier in its growth stage. And so then finally, and I think I have a few more minutes left. Yep. Turning to fungicides, we do see that our fungicides can protect us against tar spot. Just showing disease progress from 2020, but we see this similar disease trend. We started with our standard fungicide application out here at VTR1. I intentionally plant late here in northern Indiana so I can capture the disease. But what we can see is, so here we have the blue line is our non-treated, non-treated control. And then we put our fungicide out. So you can see we drop this curve here for all our fungicides. But then once these fungicides start running out of their activity 2 to 3 weeks after spraying, we do see the disease starts to ramp up in the crop canopy. So that's something we're watching and trying to monitor, how do we best lay the fungicides to get the best yield protection? And then the question comes in, should a second fungicide be applied to continue to protect it through natural senescence? And so, but I just want to share how fast this disease can develop. And so this is an example from my plots up in northern Indiana. This is 2 weeks apart generally, so 14 days difference. If you're doing a drive-by and not out scouting your corn, you will look at it and say, hey, the corn looks great, but actually we have 5% of those black spots on that leaf canopy. So you're missing it if you're not getting out there and looking for it. And 2 weeks later, you're going to come back and you're going to see, particularly if this disease is taking off, this severe blighting. And so that's what's caused the greatest concern here in Indiana, is that I've had guys that we went away on vacation, I came back 2 weeks later and my corn is burned up and it should be green for another month or so before we get ready to harvest. So that's the biggest concern, is this change in the rapid development of tar spot that can occur in that crop canopy once it gets started. And so we've been doing some uniform fungicide trials across the Midwest, and this is a summary of 8 trials that were run in Indiana, Illinois, Michigan, and Wisconsin. The good news is all our fungicides can significantly reduce tar spot. We targeted both mixed modes of action and single modes of action. So if we look at our tilt and proline, a single mode of action, yes, we can get good disease control. We're not going to market using those because we want to make sure we're providing good resistance package. And so multiple modes of action is what we're going to go for. And then we do have an assortment of products that do provide really good reduction in disease for tar spot across these multiple locations. I'm showing an example. So we had a range of severity, so across locations with high and low tar spot disease pressure. But again, we do have a lot of products that are available to try to minimize tar spot. When we look at the yields, it's a little more variable. There are 3 products that consistently did provide best yields across those 4 environments, but generally we see about fungicides protecting 1.5 to 7.9% yield over the non-treated. In our trials here in Indiana, I generally see 12 to 40 bushel yield protection depending on the season and when the disease moved in. Delaro, Revitec, and Veltima were the 3 that consistently provided increased yield over our non-treated control in these 4 various environments. We also broke apart the fungicides by their modes of action. So how many modes of action were in each of those products? And what we wanted to show is that, one mode of action does reduce disease, but if we go with 2 or 3 modes of action, we can significantly reduce tar spot severity as compared to a non-treating. And then if we look at yield benefits, the 3 modes of action was the one that consistently out-yielded both one and non-treated control, 2 modes of action falling in there, but really only 2 bushel difference between those 2 groups. So again, we do have a lot of products that are available to control tar spot. The other question comes in, how do we manage this disease? And this is one disease where we may need to be flexible on when we put our fungicides out. So this is just some timing trials that we did in Indiana from using Trivapro. From 19 to 20, I kind of put in when tar spot was first found. So 19, middle of July, 20, end of July, 2021, early July. And so that shifts which timing was optimum for managing the disease. I'm just going to show one disease curve to kind of look at what we're targeting. So again, our non-treated control, you can see this, we have this rapid exponential increase once we hit R3 of disease within the crop canopy. In 2020, when we went out with those late vegetative applications at B8 and B10, we don't do anything to the curve. They went out too early. Those fungicides ran out of activity before the disease really ramped up. If we look at the VTR2 timing, again, we shift the curve. So the fungicides were out there, they slowed the disease progress down. You start seeing them ramp up again once they've run out of their activity. When we go with R3, we missed it a little bit. It reduced disease slightly, but not as good as that VTR2 timing. And then that R4 and R5 application were too late. So you can think about the disease got ramped up. We put that R4 and R5 out, and it just isn't going to slow down the disease in that crop canopy at that point in time. We also looked at a multiple application, so going with V8 followed by VT. In 2020, that was no different from that VT curve. So that just tells us that the VT application is the optimum timing for the season. We really didn't need to go in with that earlier application. And then we've also been working with a tar spotter app with my counterpart in Wisconsin. We were still pulling in data, so it never turned on in Indiana, but it worked really well this year in trying to give us an idea when the disease was going to ramp up. And so we've been looking at, so our goal is again to shift the curve here, understanding we're slowing the disease down long enough to protect that yield for the end of the season. And so looking at the areas under those curves and trying to determine the best timing, again, VTR2 was our best timing in 2019 with no added benefit of a double application program in that season. If I look at 2020, R2 was our best timing. And if I think about it, we had drier conditions, the disease started later. It was more important to get out and have the fungicides laid out a little later to give us that little longer window of protection throughout the season. We start losing it again at R3 and R4 because the disease is ramping up, which surprised me this season though. Our tar spotter app turned on at V12, so that was a good application. My R1 had a rain on it, so it's always, you got to always monitor your irrigation and when rainfall occurs after application. So rainfall washed off some of those fungicides. We didn't get good control, but I was pleasantly surprised that we still landed in this R2, R3 window for Indiana here, at least to protecting, reducing disease in the crop canopy. So we're coming in, if my growers need to pick a timeline, VT to R2 is our optimum, but they may want to be flexible where an earlier application in this season, that V12 might have paid off with an additional fungicide application beyond that. And I think I'm running out of time, so I'm going to exit and answer any questions, if that sounds good.
Just in general for diseases, do you know of any sites, kind of like the downy mildew forecasting? Are there things like that for corn diseases that you know of?
Yeah, so it's called corn iPipe. Let me see if I can type it in the window. Let me. I search corn IPM pipe.
Okay.
If you go, and I didn't put that up, if you go to my website, it's there, but you could also, so we have the IPM pipe. It's got that national tar spot map. It would also has southern rust map right now. Those are the two ones that are pretty active with the extension pathologist across the country. So those would provide some of that. It also, if I think there's a soybean one with a soybean rust, but we're not really worried about soybean rust up in New York. But yeah, that map is there, and that's where we're tracking the disease. So if you look at it now, it shows all the counties are gray because we know the inoculum surviving year to year. And then as we become active, you'll start seeing us turn on those counties. And for my growers, it's an idea of when they need to get out and start scouting for what we're using the map for now is, yes, the disease is now active in northern Indiana. It's time to go out and look in your fields to see if you can find it, to then make a decision if a fungicide spray is going to be necessary this season.
Okay, thank you very much. So since it overwintered, this tar spot is in the soil. Do you know how long it survives in the soil for crop rotation? Can you like rotate out of a field long enough, or is it going to be there for years and years and years?
We're not there yet, and so I can't answer that. That is one of the questions. I can tell you, I know I've been in fields that it's at least surviving a couple years, and so if you have a longer rotation, we might be able to knock it down. The problem is in the Midwest, we don't have those long rotations. We're either corn on corn or corn soybeans. Right. So, and I think where I'm looking at in the Midwest, we have enough inoculum once it's going. We have so many spores blowing around that we're really not going to slow it down. It's whatever you do in your field may be optimum, but your neighbor's field may still have it, and it's going to blow around. So this disease is starting off with the inoculum in the field in that residue, but then once it gets going, we have spores moving all around in weather systems.
Do you know how far the weather systems will spread it? I mean, like miles.
Well, you can see there's one where in 2020, you can see it blew across Iowa. Yes. You can track it as he moved it across the state. So, and that's the question, we have this big question, how did it get here? If it was been in Mexico for over 100 years, why did it take so long? And so there's the question on, did we move it up with a hurricane? Finally, we had a population that could survive our winters. Did we bring it in on debris? We still haven't answered all those questions.
We could potentially have it throughout New York within, we just have it in Erie now, but it could potentially go across New York within a year or two.
Right, exactly. So it depends on, so if the weather is, so that's when you go back to some, if you look at that map of Indiana, it took a couple years before we really ramped it up. But to know if you have a bad year, like if you look at Ontario, Canada, it blew across Ontario relatively fast this last season. So I guess it just depends on how early it ramps up in your season on that western part of the state and how fast it blows across the rest. I think you have some time, but just be aware of it. Particularly in those areas that have that, so I think it's Erie County. I would be monitoring for it. And you'll see when we trigger in Michigan and Indiana, you'll see that we found it. Maybe it's time to really start keeping an eye out for it. And then I'm going to tell you, if you find it, please get a sample into Cornell to Gary Bergstrom or, the vegetable team, so you guys can document it.
And the time to scout, it looks like you were finding it in July. Is that when people should be getting out there scouting?
Yeah, so July, I think we're still not there yet on the leaves. That we know it can infect all parts of the plant once those spores are going, but that initial infection, I think we need a leaf of a certain age before we start seeing it. So that we're looking at late vegetative stages when I first find it. So
there are a couple questions that come into the chat.
Yep.
I'll read them real quick. So Julie Kickert asks, do you know of anyone who is screening sweet corn varieties for tar spot?
I think
resistance.
So I think Wisconsin's putting in some this year. They got some money for this coming season, so they're going to start working on sweet corn. Unfortunately, I haven't had any, but yes, that's going to be it's on the palette. And I guess the place to look would be Florida. I think Rich down there in Florida, you'll see turn actually relatively soon in their winter corn production. They might have some variety susceptibility in sweet corn as well. I haven't seen anything yet, but they might be one place to look. And how many day corn is in our trials? So we're 90 plus. We're looking at 90 to 110 day corn in our hybrids. So that's it. We're not using sweet corn, but we're looking at hybrids. In sweet corn aspect, I think you're going to miss it on all the early stuff. It's your late planted sweet corn at the end of the season, I think, is where you may need to watch it and how fast it's moving in.
And then Elizabeth asks about the VT applications. Are they tackling the secondary spread within a field after the initial lesions show up?
Yes, and so that's what we're trying to do is if you're scouting early enough, you can make a decision. Yes, those lower leaves may have it in a field with a history that we can make sure we need to protect that upper leaf and above. We haven't come up with a threshold, but I'm going to tell you it's game over at a certain point. So if, and I've been saying 5%, but this has not been defined yet. But if you get to the ear leaf and you have, severe, tar spot on 5% of it, those black dots, I think it's game over. We're not going to slow it down. So I've had guys that went out and tried to spray at that point and they didn't do anything to their crop, and they sprayed it a couple times. So then we're wasting our products at that point. We need to get ahead of this, unfortunately, but I think there's time. When we see it in the lower leaves, we can still put a treatment out and protect against it. But again, yes, we're slowing down that secondary cycle. And then the second application, this was in 1 year where maybe a second application might have paid for our guys to get it out there, but they had to get that early, late, the V10 to V14 application out and keep on it 3 weeks later. But this is probably the only year. Every other year, I'd say one single application is the best use of our fungicides.
Okay. And then just one last question. It will also get on the husks?
Yes. So anything on the outside of the husk. So you could pull off the husk and it's not going to be within, but those
Right,
outer
the
husks, we
outside.
can see severe infection. I didn't include any of those pictures. Well, we can see the infection. You can see the black spots on them.
Okay. Well, thank you so much, Darcy.
No
If
problem.
there's any additional questions, please put them in the chat, and maybe, I don't know, Darcy, are you sticking around?
I'll stick on and I can answer. Yes.
Okay, so thank you very, very much.
Well, hello, and thanks for inviting me. I feel a little humbled following Darcy, but Darcy, without being patronizing, that was an excellent presentation. We're still in denial a little bit as far as, I guess this last year, we maybe came out of denial, but we're way behind in research in Ohio as far as tar spots. So I really appreciate your presentation. This is an aerial shot of our north central Ohio Ag Research Station in Fremont. We've got over 100 acres there, very nice soils. We do have access to limited irrigation when needed, but quite frankly, we have very good conditions there, and it makes it an awfully nice place, let's say an easy place to do variety trials. Although I will admit, it's basically as nice of atmosphere as we have, or climate as we have in Ohio. So basically, when I look at my variety trials, I have to clarify with people, look, under ideal conditions, this is what you can expect. But that being said, it's a great place to do research. I'm in a neighboring county. Just to give you an idea, Fremont is just south of 80 and 90 run together where we are. And just at the end of the Sandusky Bay of Lake Erie. So it's farm country all around, as you can see, very nice Coalwood soil here. Well, basically a variety trial is just grunt work, I will say. Luckily, we have access to some labor, usually college students, some high school students, and they help me tremendously because it takes, well, I use 8 people in my trial the days we're harvesting. It's usually several hours a day to get through it all hand harvest. And well, it's just a lot of grunt work, quite frankly. It's not fancy, but what makes it, the uniformity of it gives us the validity. That's the only value of a variety trial like this, is if you can keep it uniform and give each variety an equal opportunity to shine, if you want to look at it that way. Again, in our situations, we have four reps. Last year we had 43 varieties, all SH2s. And why SH2s? This started 15 or 20 years ago already, and I've done it the last 8, I believe, as far as this trial. 20 years ago, I worked with commercial sweet corn growers as well, who are shipping all over the country. They were using SH2s, but our roadside markets never used them. And in fact, back then, I've been with Extension 16 years. 16 years ago, when my commercial growers gave me sweet corn, quite frankly, I would not eat it because it was too much like store-bought and corn to me, too crunchy, if you will. Of course, the SH2s have come a long way, leaps and bounds. And as far as tenderness and eating quality in general, just excellent. Last year, we had 43 varieties, which is more than I ever want to do again. because like I say, we have 4 reps of each one, so it's just a little too much to take that many varieties. We're going to limit it to 35 this year. When we started doing these trials, though, part of the idea was, how did this corn store? We've kind of gotten away from that. We used to store it for a week and then sample it and see how it tasted after a week in cold storage. Hey, it's still a week old corn. I don't care. We don't look at it that way anymore. We do taste test it fresh. We do know this whole the SH2 family, all of those will store better than, say, normals or SEs, and that's kind of a given now. So we do no longer do the 1 week storage, but I think we gather enough information to give producers a handle on what will be saleable for a few days, maybe not a week out. And I don't promote selling week old corn, so I'm fine with that. Again, just grunt work. We're having hand harvesting. This is our former manager. He left it this year after many years, Matt Holflick, and he's been great to work with over the years, but just retired a month ago. This gives you just a little view of our station there. We bring it up on wagons. First thing we'll do is get photos of it, both stripped and in the husk, just so we have a long-term record of it. A lot of times if I'm doing local meetings, people will ask me to see it, and so I will have a record of each one. I don't have those available today, frankly, but so we just take a picture of 5 years. And when I said we had 4 reps, we harvest all of them, and in those 4 reps, there are 4 rows. We harvest the center 2 rows of each rep, and we use all 4 of them, all 4 of the reps to get our indication of yield. I'm sorry, but we only pull 2 of them and sample those as far as our characteristics that are in my report. And just another picture of, well, what we do when we bring it up, take a picture of it. All right, my first table here, table 1, shows variety characteristics, basically emergence. And this is one thing, we only make comments on things that don't come up evenly, or if there's a problem, we'll make a note. Other than that, we don't show too much on it. But on my first chart here, of course, color characteristic, but then this is somewhat important locally. It's good local knowledge. We always list the variety, or I'm sorry, the maturity as listed by the breeder or by the seed company that supplies it, and then what it actually did for us. And in our case, we are looking at maturity from the date it was planted till the date it was harvested. Some places do it a little differently, but I point that out. Most of the cases, most of the time, since we do it that way, our observed maturity will be longer than listed, but not under ideal circumstances. If we get very nice growing conditions, we'll sometimes beat the listed maturity. So, and we also have harvest date on there just to give you an idea. Table 2 then, and hopefully this is available to you. I did send it along, so I think you can link to it if you're interested in my trial from last year. What I'm showing on the screen is just, well, in this case, 17 of the 43 varieties. So I just gave you a snapshot. But in table 2, we look at things that we would measure out in the field. So ear height being a big one. If you don't think ear height matters, you've never harvested sweet corn, obviously. But we like to give some indication of what we found. And again, this is under ideal conditions. So truthfully, the value here is that under those conditions, what's the ear height? Then you can kind of compare one to another, because we do try and be very uniform in our measuring and such. Basically, what I find, anything under about 20 inches, we have some problems. And it's not just harvest problems, although it makes it inconvenient to be down in the 16 and 18 range. We'll have a lot lot more pressure from not just raccoons, but if it's that low, also from groundhogs. Anyway, ear height does matter, no doubt. We have actually 2 people harvest all of our plots. And so I feel fairly good about coming up with an indication of snap. On ours, a 3 rating is basically average, excuse me. A 3 rating is average. The higher, the easier the snap is. And you can see most of them are fairly close to average, but some are real boogers to pick. And we try and indicate that. And if they have a 2 rating, it's pretty tough to pull. We have harvested dozen, marketable dozen on that as well. Basically, that used to be one of my main goals, is to look at yield. As far as I'm concerned, many things enter in there, but mostly cultural and environmental factors enter in there more than the variety itself, perhaps. So what I try and do there is just monitor any problems. If something absolutely can't yield with the others, we try and make that known. But when I talk to audiences, I pick a number. If you can come close to the 2,000 for what we're planting, if you can come close to the 2,000 dozen per acre, I would just label it as adequate and check that off the list. There's just a lot of factors that enter in. The other thing too, we harvest every marketable ear. My commercial growers go crazy because they harvest 1 ear per, so they really don't care to see that we actually have more marketable dozen than our population out in the field. When they see that, they discount my work altogether. But really, I just ask people, as far as harvest or yield, that is, please just say yes, it's adequate, or no, it's not. And quite frankly, most varieties are quite adequate of producing the 2,000 dozen per acre if conditions are right. Table 3, as far as things that are affected by environment and other cultural practices, a lot of these things on table 3 are, we look at husk cover, flag leaves.
Mike,
Yes.
I'm sorry to interrupt. It looks like your slides might not be advanced.
Oh, I didn't advance today.
I'm sorry. I'm sorry.
Sorry about that.
Thank you to Elizabeth for noticing that.
Thank you. Yes, thank you very much for alerting me to that. I'll hop back to table 2 just so you can see that a little bit. And I went the wrong way, didn't I? No, I didn't. There's table 2. And then table 3 that I'm talking about now, we look at husk cover, flags, and basically that's just appearance, the flags. But it does make a difference in sales sometimes. Husk cover, very important to us. We have bird problems. The less husk cover we have, the more likely we are to have bird damage. But what we found in the last few years, if we get moisture right at harvest time, we'll have corn growing right out the end of the husks if we don't have adequate husk cover. And that's from a saleability standpoint. Sometimes people don't like to see it showing at the end, and there's oftentimes insect or bird damage if you do have exposed kernels. So we give it an overall husk rating. That's pretty much appearance. And shank rating. This is big for our shippers, especially. They do not want the huge shank on it. And most of them are, well, there is some variety in that. Roadside markets, they'll put up with a shank, but it usually requires a little more handwork to get that shank off. So we do like to list that. If something sticks out, at least it's something to consider being a problem. Tip fill, from time to time, I thought about even taking this off the list because it's so much affected by pollination and other environmental factors, frankly. But we leave it in there. Some varieties are more prone to not fill the tips, but in general, I think environmental conditions have more to do with that than anything. Rowing or rows are fairly, we'll say, heritable, I guess. you can pretty well predict how many they will have, although you see under good conditions, even a corn that will usually have 16 rows may have 18 or even more if thin in population or something. Rowing can be affected obviously by pollination, but some varieties are very apt to have haphazard rowing, and that can be a problem. If we sell it in the husk, it's fine, but it's not as acceptable when the consumer pulls that back and we have rows that are not straight. On here, we're looking at color. That's basically whatever color it is a vivid color. And then we measure the length and the girth of the ears, which is kind of self-explanatory. Lastly, and this one's kind of big, and it also is open to some scrutiny. I will say, as far as uniformity, I try and use the same 5 people as testers the entire year. And I can say I make a lot of corn snobs out of some of my young help. I consider myself a corn snob. If I'm going to eat corn, life's too short to eat bad corn. So I like it tender and I like it sweet, but I want it to have a little corn flavor as well. So anyway, we do our best to go through 43 varieties and be fair about it. Basically, as far as tenderness, the average is a 3. Now, I will say that average has moved over the years. We have much more tender SH2s than we used to. And this can vary a little bit by maturity, and we try and be fairly uniform in our maturity as well. That's one of the toughest parts about this project is harvesting at peak maturity. And we usually use a group of 3 of us to scout. We go by what we would expect from maturity tables, but we look at them and say, okay, this one's at peak maturity today. So even though they're within 7 days, we do tend to have everything harvested in that plot within 10 days. So they vary a little bit, but tenderness and a 3 average is still pretty acceptable, I will say. Like I say, we are tough on our grading, but 3 still acceptable. Frankly, a 3-3-3 here, as far as tenderness, sweetness, flavor, is a pretty doggone good corn. If it's below that, if it's 2.5 in any of those categories, still pretty decent. Below that, we've got some problems, and it tends to be what my help would typically call chalky corn. If you think back to the original SH2s, I'll call them store-bought corns, because if I went to Walmart, that's what I would expect to get, 15 years ago. If you take one bite and it's a little bit tough or overly crunchy, and you get that chalky flavor, my group will usually not give it a favorable score at all. So if you can look at this, one that shined through for you, well, of course, Nirvana has a pretty good track record, well above average in our trials pretty much every year that it's been in. Solstice came along a few years ago for an early corn. That one kind of got my attention right away. I didn't say it before, but I do these all blind. I never even look at what the variety is until we're all done. I strictly go by the number. Of course, they're all randomized, but anyway, we do it by the variety. And when it's all said and done, then I add the variety name in. I think it's a fair way to do it. But when we're doing it, if I find one that I find exceptionally good on that given day, I'll go back to the record and find out what it is. But I do try and do them blind. When Solstice came along, it got my attention, I will admit. For an early corn, it just had very good flavor, tenderness and sweetness, and I really liked it. Then I raised it on my home farm, and I will say at the station, they do a better job than I can do myself. They just have, maybe the soil's a little better, but the sweet corn gets the utmost attention. So it's really ideal conditions. When I raised Solstice at home, what I found was we had indications that the the ear was fairly low, but under any stress, we were getting ears very, very low, to a point where it was a real problem. And we had problems with raccoons in it. But I do like Solstice under good conditions. It's high enough ear placement, and I hate to single that one out, but it's just one that came to mind because it got my attention a few years ago when it came out. But you put it on your home farm, sometimes it doesn't react exactly the same. So really, that's all I have. This is another picture of the home place. I have contact info here, and be glad to talk to anybody. That is my county office number. As you can tell, I am in here, county office. You might be able to tell that from my 1950s nice paneling in the back, but we're in the northern Ohio and right in the middle of the state. My phone number's there and my contact by email, and be glad to talk to anybody. I'm always willing to send out this trial. I should give credit to the OPGMA. We have a vegetable checkoff in our state, which is voluntary, but they raise a fair amount of money, and most of it goes back into research grants on things like this trial. Most of them are fairly small grants, and in our case, we get $3,000 a year from them that goes to to help offset the research. But I do appreciate their support, and they found this to be a worthwhile project for the last 8 years now. So thank you for your time. If I can answer anything, or if you're interested in the report. One thing I have seen this year, the value of it perhaps is it allows you to compare things when one seed is not available, because we run into that all the time with a variety being short. Sometimes you can look at a report like this, raised under the same conditions, what's a good replacement for that product? And I think that's how most of my producers would use it.
Thank you very much, Mike. So Mike's report, as well as Darcy's PowerPoint presentation, the PDF of that will be posted to our proceedings at the Producers Expo website. So if anybody wants to look at those tables a little more closely, they can go there to look at those. Are there any questions for Mike? I just had one quick one. I know if you want to stop sharing, then we can put up the QR code, but I just had a quick question. Have you noticed, I know you're not evaluating this, but any disease differences on certain varieties?
For the most part, no. But we do monitor closely, but no, we don't. We haven't in the past, I should say. 20 years ago, they did more of that. Now, typically we can avoid, since we're planting fairly early, we tend to avoid most of the leaf diseases. In years past, when we've had a lot, any pressure from particularly leaf disease, we've made note of it on specific varieties. Mm-hmm. Other than that, though, it has to be a fairly obvious problem before I do make a note of it.
And you're not treating it? Are you treating it for anything? Like if the disease pop up?
Typically we do not spray it with a fungicide. We aren't above doing that, but most years we do not find that we need to. I know a couple of years ago we had a particularly wet period, a month before harvest, and we were starting to see some northern particularly, and it came early enough that we did treat, but typically we do not.
All right. Well, thank you very much. Thank you both to Darcy and Mike for the presentations, and to all the participants, as well as Kim, who's been kind of working behind the scenes. I also want to just note that this presentation has been recorded and it will be available on the SIPS YouTube site. So if you want to go back and view it, that'll be available. So
This has been a production of Cornell University, on the web at cornell.edu.